Hello my dear student, welcome to the MS Science Academy. Myself Meenu Srivastav, and today we are going to discuss about the biological significance of cholesterol and conversion of cholesterol into bile acid, steroid hormone and that vitamin D. Okay, so first we will discuss about the structure of cholesterol. The basic structure of cholesterol is like that, and its nucleus, its a basic nucleus is cyclopentane per hydrophenanthrin nucleus. Cyclopentane per hydrophenanthrin nucleus, okay, and here the cyclopentane, this is the pentane ring, and this molecule, this ring is the it's a per hydrophenanthrin nucleus. Okay, so the basic nucleus of the cholesterol is cyclopentane per hydrophenanthrin nucleus, and the molecular formula is C27H46 and O. Okay, so here the cholesterol is very much important for the synthesis of different like the steroid hormone, vitamin D and the other and the bile acid formation. Simultaneously it provides the fluidity and fluidity to the cell membrane, and it provide the we can say it provide the it make easy the transport mechanism through the cell membrane. Okay, so now come to the biological significance of cholesterol. It is a precursor for the synthesis of steroid hormone. It is a precursor for the synthesis of steroid hormone, bile acid and the vitamin D. It is a basic structural component of the cell membrane. As we know that the cell membrane composed with the protein and lipid, and it also has the cholesterol. So the cholesterol provide the stretchability, fluidity and it make easy the transport through the cell membrane. Third, it plays a major role in the digestion and absorption of lipid. Actually, the cholesterol convert into the cholic acid in the liver, and that cholic acid is very much essential for the absorption of lipid. So here the cholesterol plays a major role in the digestion and absorption of the lipid. Especially, it serves as a precursor for the synthesis of steroid hormone. It serves as a precursor for the steroid hormone like the glucocorticoid, mineralocorticoid, androgen, progesterone and the estrogen. Okay, it serves as a precursor, basic precursor. It serves as a because it has the cyclopentane per hydrophenanthrin nucleus, which is the basic structure for such steroid hormones. So cholesterol provides such basic structure or nucleus for the synthesis of steroid hormone. Okay, it is required to transport the plasma membrane. As we know that the plasma membrane is having lipid, protein and cholesterol basically, and this basically that protein and the cholesterol provide the spatial transport mechanism for transport to make the transport easy in the easier way. Okay, it serves as the precursor for the synthesis of oral contraceptive, because in the oral contraceptive the main content is the progesterone and the estrogen. So as just now we have seen that it has the precursor for the synthesis of progesterone and estrogen. So in the oral contraceptive the same combination they are providing, so it help to form the or to synthesize the oral contraceptives. Okay, it provide the next point is that it provide the fluidity of the membrane. Fluidity means it provide it make easy transport through the membrane. So basically in the biological term the transport mechanism, or we can say the go through mechanism, is known as the fluidity. So it provide the basic fluidity to the membrane, or we can say it make the transport easy in the through the cell membrane. Okay, it function as a insulating cover for the transmission of electric impulses in the nerve tissue. Okay, so in the neurons, as you I don't know you know about the nervous tissue. The nervous tissue in the nervous tissue the basic structure of the nervous tissue is the neurons. Okay, so this is the basic structure of the neurons, and neurons during the electrical impulses, as the electric impulses go through, so these impulses jump towards through the neuronal membrane, or we can say neurolemma. Okay, so the cholesterol provide the insulating covering. The cholesterol provide the insulating covering, so such ion cannot be leaked out from this membrane. Okay, so if such ions, sodium and potassium ion, leaked from this membrane, the potential impulses will reduce. Okay, so to protect from the leakage, this cholesterol make the insulating covering. Now this is whole for the biological significance. Now come to the conversion of cholesterol into the bile acid, vitamin D and steroid hormone. Now come to the synthesis of bile acid from the cholesterol. The synthesis of bile acid is started in the liver and it completed in the intestine. Okay, so primary bile acids are formed in the liver and the secondary bile acids are formed in the intestine by the action of intestinal bacteria. Okay, here the cholesterol is converted into the 7-hydroxycholesterol in the presence of 7-alpha hydroxylase enzyme, and in this process the NADP is generated. Okay, so 7-hydroxycholesterol is converted into the cholic acid and chenodeoxycholic acid. The cholic acid and the chenodeoxycholic acid are the example of primary bile acid. Okay, now the cholic acid by the conjugation with the glycine is converted into the glycolic acid, and this cholic acid by the conjugation of taurine is converted into the taurocholic acid. Okay, so the glycolic acid and the taurocholic acid is also the example of the primary bile acid. Now the chenodeoxycholic acid by the conjugation of taurine and the glycine convert into the tauro or glycochenodeoxycholic acid. So the tauro or the glycochenodeoxycholic acid is also the example of primary bile acid. Now this part is completed in the liver, but after this next part is going towards the intestine, and the intestinal bacteria will work on this and convert into the secondary bile acid. Here the glycolic acid and the glycolic acid is converted into the deoxycholic acid, and this deoxycholic acid is the example of secondary bile acid, and tauro or glycochenodeoxycholic acid is converted into the lithocholic acid. And this conversion, deoxycholic acid and the lithocholic acid formation deoxycholic acid and the lithocholic acid formation takes place in the intestine in the presence of intestinal bacteria. Okay, so deoxycholic acid and the lithocholic acid are the example of the secondary bile acids. So that's why in the starting I have said that the primary bile acid conversion is in the liver or formation in the liver, but the final secondary bile acid formation occur in the intestine. Okay, so here the primary bile acid convert into the secondary bile acid by the help of deconjugation and the dehydroxylation reaction. Okay, so primary bile acids and the primary bile acids formation occur till the liver, and the secondary bile acid formation occur in the intestine. Okay, so this is whole for the complete synthesis of the bile acid. Now we will study about the synthesis of steroid hormone and the vitamin. Now come to the formation of steroid hormone. Okay, here the cholesterol is a precursor for the synthesis of, as we know in the biological significance we have studied that the cholesterol is a precursor for the synthesis of 5 classes of the steroidal hormone like glucocorticoids, example cortisol, mineralocorticoids, example aldosterone, progestin, example progesterone, and the androgen, the example is the testosterone, estrogen and the estradiol. Here now come to the cholesterol, come to the synthesis of the steroidal hormone. Here the cholesterol is converted into the progenolone. Okay, the cholesterol is it's a 27 carbon, as we have studied the structure is C27 H46 and O. Here the cholesterol is converted into the progenolone. The 27 carbon molecule is converted into the 21 carbon molecule, and this progesterone convert into the progesterone into the 21 carbon molecule. Here the progesterone is convert into the, as the progesterone is the is under the class of progestin. So here the progesterone is converted into the cortisol. Cortisol comes under the category of glucocorticoid, which is a 21 carbon basic molecule, and the aldosterone comes under the category of mineralocorticoids, okay, so which is a 21 carbon molecule, and the estradiol is the containing the estrogen 18 carbon molecule. So here not you should not go should not go into the very much deep, because it's not coming as a long question, maybe it's coming as a short notes, okay. So this much matter, I think it's more than sufficient, okay. So now come to the study of the synthesis of vitamin D formation. Now come to the synthesis of the vitamin D. The vitamin D is synthesized by the 7-hydroxycholesterol, which is the derived or the previous precursor form. We can say the precursor form of the cholesterol. The precursor of the cholesterol which is present in the skin is 7-hydroxycholesterol. Okay, so it means in the skin the cholesterol is present in the form of the 7-dehydroxycholesterol. Okay, so in the skin, the ultraviolet rays, when they interacted with the skin, the 7-dehydroxycholesterol is converted into the cholecalciferol, which is the vitamin D3. Okay, now this cholecalciferol is converted into the 25-hydroxylase, and this 25-hydroxylase, in the presence of 25-hydroxylase, this cholecalciferol is converted into the 25-hydroxycholecalciferol. This 25-hydroxycholecalciferol is converted into the 25-dihydroxycholecalciferol, okay, in the presence of 1-alpha-hydroxylase in the kidney. Okay, so this conversion, this dihydroxy conversion is known as a calcitriol, which is the form of the vitamin D. Okay, so this whole form involve the skin and the kidney. Okay, so this is whole for the formation of vitamin D. Okay, so I hope you have enjoyed my lecture. Any doubt, kindly ask in the comment section. Otherwise, please like, share and subscribe. Thank you.